Good morning. For today, we will have a lecture on lipid metabolism. When we talk about lipid metabolism, it's either we make the fat or we break down or activate or oxidize the fat. So for today, we'll have this fatty acid synthesis. So carbohydrates, protein, and other molecules obtained from the diet in excess of what our body needs can be converted to fatty acids, and they are stored as triacylglycerol. So with biosynthesis of fatty acids, there are specific characteristics, no? So it is synthesized in the extra mitochondrial system or in the cytoplasm. The process involves incorporation of carbons from acetyl coenzyme A into a growing fatty acid chain. So when you say biosynthesis of fatty acid, it refers to the complete synthesis of a 16-carbon palmitic acid from just 2 carbon molecule of your acetyl coenzyme A. So again, it is extra mitochondrial. That's why it's located in the cytoplasm. And synthesis of fatty acids occurs in almost all organ systems, particularly in the liver, in the mammary gland, and in adipose tissue. So for each process, every process, there is usually a cofactor requirements. For biosynthesis of fatty acid, it requires the following cofactors: the NADPH, ATP. So therefore, your synthesis requires an energy. Okay? Manganese is also one of the cofactors, biotin and bicarbonate. The immediate substrate is an acetyl coenzyme A, and our aim in its process is the free palmitic acid, which is a 16-carbon palmitic acid. Okay, and in every process, there is always a controlling step, the regulated step, which is the production of malonyl coenzyme A. Malonyl coenzyme A is a 3-carbon molecule. So this fatty acid biosynthesis requires the acetyl coenzyme A. So where does acetyl coenzyme A come from? If you can't remember your carbohydrate metabolism, this is made from the pyruvic acid. Okay, so meaning to say it came from your glycolytic pathway or your glycolysis. Okay, but where is the location of your acetyl coenzyme A in your carbohydrate metabolism? After it goes towards the process of glycolysis, the acetyl coenzyme A, after it's being produced from the pyruvic, is located in the mitochondria. And what did I say? The fatty acid synthesis occurs in the cytoplasm. So therefore, your acetyl coenzyme A needs to enter the cytoplasm, and that is in the form of citrate. And that is catalyzed by the enzyme citrate lyase. Okay, so when you say fatty acid synthesis, it requires cooperation of several metabolic pathways. Okay, so you will be able to understand, appreciate fatty acid synthesis if you can still remember what happened during your glycolytic pathway. Okay, so this is how the fatty acid metabolism and carbohydrate metabolism overlap. Okay. So aside from overlapping with carbohydrate metabolism, it also overlaps with protein metabolism. Okay, so therefore, never forget all the lectures you had when you like, for example, you're done with your carbohydrates, now you're with fat, and then when you go towards protein metabolism, everything is interrelated. Okay, so there is an overlapping with all this metabolism. Okay, so I think you remember this particular slide. Okay, this particular figure. So this is your glycolysis. Okay, so glucose is there. So your glucose, and then now you have your pyruvate, and your pyruvate enters the mitochondrial membrane, wherein it is being catalyzed, or yeah, it's being catalyzed by the enzyme pyruvate dehydrogenase to form the acetyl coenzyme A. Remember what I said, your acetyl coenzyme A is the initial substrate in your fatty acid synthesis. Okay, but what's the problem now is that your your fatty acid synthesis is located in the cytoplasm. But where is your acetyl coenzyme A? It's inside the mitochondria. So therefore, it needs to move out from the inside. Okay, it needs to move out from the inside, moving out from the mitochondrial membrane. Okay. Okay, you wait because I need to with my pen, so you can you will be able to see it. Okay, I'll use the red. Okay, so this one. Okay, so here your acetyl coenzyme A. So this is your acetyl coenzyme A. It needs to move out from the cytoplasm, okay, as citrate. Okay, so here, so it's a citrate. No, it needs to move out from the cytoplasm as citrate. And once inside the once in the cytoplasm already, it is being catalyzed by the enzyme ATP citrate lyase. And this particular enzyme will it will cleave the citrate into acetyl coenzyme A and oxaloacetate. Okay, and of course, your oxaloacetate will go again towards the glycolytic pathway. But what we are concerned right now is this one, no? Wherein your acetyl coenzyme A will proceed to this process, forming the 16-carbon palmitic acid. Okay, so here, once your acetyl coenzyme A is here, it is being carboxylated. Okay, this one, this is the very important enzyme, which is your acetyl coenzyme A carboxylase. So there would be carboxylation. Carboxylation meaning to say there is an addition of a carbon molecule. So therefore, it utilizes one molecule of carbon dioxide and also an ATP. Okay, it requires this energy to form the malonyl coenzyme A, which is the initial and controlling step in the formation of your palmitic acid. Okay. 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 So as part of the requirements or the cofactor requires in which are required in fatty acid synthesis, it's your NADPH. So your NADPH is sourced from three major enzyme system. The one that is involved in the pentose phosphate pathway, the malic enzyme, and the isocitrate dehydrogenase. So I'll go back to this slide. Okay, so this one is one of the major sources of your NADPH. The isocitrate dehydrogenase, and then also the malic enzyme. Okay. Oh, sorry. Okay. So these are the two enzyme systems that are involved. Okay, so your acetyl coenzyme A carboxylase and the one that is responsible for the condensation, for the formation of the 16 carbon palmitic acid is the one that has 7 enzymes. That is what you call fatty acid synthase system. So going back, the 3 stages of your fatty acid synthesis. First, what we did, we transported the mitochondrial acetyl-CoA to cytosol. Okay, I hope at this point you can still remember how this acetyl coenzyme A moved towards the cytoplasm, okay, as the citrate. Okay, and then the second step is the carboxylation of acetyl-CoA to malonyl-CoA by the enzyme acetyl coenzyme A carboxylase. And this is the regulated and controlled step. And then the third one will be the assembly of your fatty acid chain by your fatty acid synthase. Okay, so this is how it's being transported. And then formation of your malonyl coenzyme A after it's being carboxylated. Biotin there is one of the cofactors and also the bicarbonate. So with the carboxylation of your acetyl coenzyme A, it is being regulated both short-term and long-term. The short-term regulation of that enzyme is the enzyme undergoes allosteric activation by the citrate. Another regulation is by reversible phosphorylation by the counter-regulatory hormones. So with the phosphorylation, the enzyme is inactivated. So therefore, if it's being phosphorylated, particularly with the presence of your glucagon, no, and your counter-regulatory hormones, the enzyme will not be active. So there will be no fatty acid synthesis. Whereas if with the presence of insulin, the role of insulin will dephosphorylate the enzyme. A dephosphorylated enzyme will make it active. So there would be more formation or synthesis of your fatty acid. Okay. So this one, your acetyl coenzyme A becomes dephosphorylated with it becomes active. Okay, if it's phosphorylated, it becomes inactive. If it is dephosphorylated, it becomes active. So there will be more malonyl coenzyme A here. Okay, the one that phosphorylates your enzyme are your counter-regulatory hormones such as your glucagon, epinephrine, and so on. Okay, so here it shows that the insulin, so there's a plus here. So insulin stimulates this enzyme. So therefore, with the presence of insulin, there would be more fatty acid synthesis. So if you just imagine having a patient with uncontrolled diabetes, okay, uncontrolled. So what's the problem in diabetes mellitus? There's no insulin or deficiency of insulin. So the body of these patients with uncontrolled diabetes, they are more thin, okay, because they don't have the insulin. They don't have the power to synthesize fats, because what predominates in diabetes mellitus are the counter-regulatory hormones such as your glucagon. Okay, so another way of regulating the acetyl coenzyme A carboxylase is through long-term regulation. Definitely, is because of the prolonged intake of high caloric and high carbohydrate diets. So this can increase the synthesis of the enzyme acetyl-CoA carboxylase. So the higher intake of this kind of products or food products may promote fatty acid synthesis. Okay, so after we transported, after we carboxylated, then let's go now to the assembly of the fatty acid chain by the 7 enzymes involved in the fatty acid synthase system. Okay, the first thing that will happen is there would be loading of your acetyl-CoA and loading of a molecule of your malonyl coenzyme A into acyl carrier proteins. So acyl carrier proteins are very important in the assembly of your fatty acid chain. So during that process, there would be 4 reactions. Okay, first is condensation of your acetyl ACP and malonyl ACP. So what did I say? Acetyl A or the acetyl ACP has two carbons. Okay, malonyl coenzyme A or malonyl A has 3 carbons. This is a product of the carboxylation of your acetyl A. No? And then during the condensation, there would be removal of 1 carbon. So there would be condensation. There would be decarboxylation that would happen, no? So therefore, if there is removal of 1 carbon, so there would be acetyl malonyl ACP complex here, no? And this 4, okay, this particular compound will undergo reduction. After that, it will dehydrate and then it will be reduced again. Okay, so this one is the fatty acid synthase complex. So as I mentioned, there are 7. No, there are 7 enzymes wherein they are involved in the biochemical reactions after the condensation. No, and then the last one, the 7th enzyme, which is the thiolase, is the one responsible for the release of the formed or the final product, which is your palmitic acid. Okay, so I mentioned this already. Okay, so your acyl carrier protein. So this is your acyl carrier protein. This is your and then one of the vitamins that would act as a key player or as a cofactor is your pantothenic acid. Okay, so look at this particular figure. Okay, so this is your ACP. No, this is your ACP, and this is your this one. The first one is your acetyl coenzyme A. Okay, wait. Okay, so this is your acetyl coenzyme A, and then this is your malonyl coenzyme A. So they will be they will condense. Okay, they will condense. So this is 2 carbon. This is malonyl coenzyme A. Okay, so I'll show it with the pen. Okay, okay, we'll just have the arrow. Okay. Okay, wait. We have the color. Okay, so this is your acetyl coenzyme A, and then this is your malonyl coenzyme A. Okay, so they will condense, now forming the 5-carbon acyl malonyl enzyme. Okay, and then as I said, it will after condensation, this one is 5. So after condensation, there would be removal of 1 carbon, forming now this 4-carbon, 3 ketoacyl enzyme or your acetoacetyl enzyme. Okay, after which it is being reduced by your 3-ketoacyl reductase, forming this hydroxyacyl enzyme. And then it will be dehydrated, okay? And then forming the unsaturated acyl enzyme. And it will be reduced, forming this acyl enzyme, which is a 1, 2, 3, 4 carbon molecule. And what is our aim? We need to produce a 16 carbon palmitic acid. So therefore, this particular cycle, okay, will go back again, okay, wherein it will condense with a molecule of malonyl coenzyme A. So for every repeat, for every cycle that it undergoes, there would be an addition of 1 molecule of malonyl coenzyme A. Okay, so therefore, this one, okay, okay, how can I forward this one? Okay, so, okay, so loaded. So there's loading of malonyl to fatty acid synthase. So condensation, reduction, dehydration, reduction. Okay. And then, so this is how your carbon dioxide is being lost. Okay. So we have this. Actually, I've mentioned this already. No? We'll just move a little forward. And then after that, okay, after that, the next cycle begins. Another malonyl group is already linked to another ACP and combines with a 4-carbon molecule that has been formed during the first process. Okay, so this one, your acyl, the acyl enzyme goes back again, okay, condenses with malonyl. So therefore, this sequence is repeated 6 times, okay, so that you can have a final product, which is a 16-carbon free palmitic acid. And then the 7th enzyme in the fatty acid synthase system, which is the thioesterase, will be responsible for the release of your free palmitic acid. Okay, so that is the process of your fatty acid synthesis. So the next question is, what will be the fates? What will happen to the free palmitic acid that has been formed? Okay, so first, it can be utilized to esterify, okay, for further esterification into acyl glycerol. Okay, it could be, it could make up a triacyl glycerol. And remember, your triacyl glycerol is the storage form of fat in humans. The free palmitate also can be used for chain elongation or desaturation, and it can be esterified to cholesterol ester. So this is the one that I said that any excess of the sugar, any excess of the glucose will be formed into cholesterol. Okay, so this is a simple representation of your synthesis of the fatty acid. Okay, by just looking at this particular slide, you will be able to know, to identify 4 answers to a question. 4 answers to 4 questions, I mean. Okay, first question is, what is the initial substrate in fatty acid synthesis? The answer is acetyl coenzyme A. And what is the initial and controlling step? It is the formation of a malonyl coenzyme A. And what is the end product? It is the formation of your palmitic acid. And what is the important enzyme in the fatty acid synthesis? Is your acetyl-CoA carboxylase. Okay, but just remembering the slide, then you will be able to get 4 correct questions. Okay, so this is the stoichiometry of the overall synthesis of palmitate from acetyl and malonyl coenzyme A. So we utilize an acetyl-CoA, 7-malonyl-CoA, 14 NADPH during the process, and also 14 hydrogen. And we release palmitic acid because of the decarboxylation during the condensation part. So we released 7 carbon dioxide, 6 water, 8 coenzyme A's, and 14 NADP. So how is lipogenesis being regulated? The main factor definitely is nutritional state. Any excess carbohydrate, all the metabolites of the carbohydrate metabolism, your pyruvate, lactate, and acetyl coenzyme A can be stored as fat. Of course, the rate is very high in well-fed state, and it's depressed in diabetes mellitus because of the absence of insulin. And of course, your acetyl coenzyme carboxylase is considered as the most important enzyme in this process, and it is activated from an inactive dimer to an active polymeric form by citrate, and it is regulated by these hormones, glucagon, epinephrine, and insulin. So you all know already how the insulin stimulates lipogenesis. First, it increases the transport of glucose into the cell, particularly in the fat tissue or in the adipose tissue. It increases the availability of both the pyruvate for the fatty acid synthesis and even the glycerol 3-phosphate for esterification of the newly formed fatty acid. So the glycerol 3-phosphate is the backbone or the initial backbone in the triacylglycerol esterification. And also it converts the inactive form of pyruvate dehydrogenase to its active form in the adipose tissues. So it has the ability to depress the level of intracellular CAMP, thereby inhibiting lipolysis, reducing the concentration of plasma-free fatty acids and long-chain acyl coenzyme A. Okay, so therefore, as it decreases the cyclic AMP, lipolysis is inhibited. Therefore, plasma-free fatty acids are decreased. Okay, so again, showing you this one, the role of insulin in our fatty acid synthesis. So again, the one that's plus there, therefore, insulin promotes the formation of your acetyl coenzyme A, thereby increasing the rate of your fatty acid synthesis. And your glucagon, particularly, all the other counter-regulatory hormones will suppress the activity of this enzyme acetyl-CoA carboxylase. So therefore, this one, the glucagon, will promote lipolysis. Okay, so that ends my lecture on the fatty acid synthesis. Thank you so much for your attention.